This is Audio Munity, a podcast about our body's never-ending fight with the outside world. Fun. Hi everyone, this is Audio Munity, episode... God, what episode is it even? 52?
52, yeah. I think it's 52.
Recorded on January 30th, 2026. First recording of 2026. I'm Kevin Bonham, Tufts Medical Center, joined by Matt Woodruff.
Hey, everybody.
And this episode today is brought to you by Postnasal Drip and like 10 feet of snow that I felt like I dug out from last weekend. How have you been, Matt?
Yeah, okay. Weather's tough here. It's just below freezing, which sends everybody into a panic.
Yeah.
It may snow a little bit, and so the
Goodness.
entire city, has proactively canceled itself. We
Cool.
had last weekend threat of icy rain, which, to be fair, freezing rain can be pretty.
Yeah.
We shut down all the schools, basically every business, for a 43-degree rainstorm that immediately sort of drifted off. So that is how Atlanta handles weather. We have the weather, and then we just trust that nature will eventually take it away.
Cool. Yeah. When I stepped outside my door today, it was 1 degree Fahrenheit. Felt like minus 17. And the commuter rail was canceled because apparently tracks don't function when it's cold.
Right.
And everybody was freaking out. So, we haven't been doing the weather lately on Audio Munity, but it feels very relevant. Today, we're going to talk about a paper that purports to describe a new cell type called an adenopage. Query whether that actually makes sense. I may have some things to say about that.
Yeah.
But the big announcement that I think we want to make here in the new year is that we have decided to move from the bespoke website and artisanally crafted RSS feed to Substack. So the idea is we're going to move everything over there to Substack. And there's a few reasons for this. So the first reason is that what I have been doing when I edit these episodes, the platform that we record on, Riverside, is really great for doing a basic edit. They produce transcripts and you can highlight text and cut out text and it'll cut the corresponding video. Makes my life much easier than back 10 years ago when I was using Adobe Audition to manually edit all these things. But then what I've been doing after I do that edit, I use another software to edit and put in the music at the beginning and the end, composed by Rachel Reinick, by the way, and then export a video. And then I've been manually converting that video to audio. I have been writing up the show notes for the web page, which I then copy to archive.org, where I upload the audio, which I then copy to Patreon, where I upload the video, and copy to YouTube, where I upload the video. And so all of these individual steps are just a lot of extra work. With Patreon, I can upload the video. It automatically generates a transcript, generates short clips, posts to YouTube, makes the audio feed. So all of that is just much, much easier. And I think the other piece of it is that Matt and I have been talking off and on, sometimes on recording, sometimes off, about doing a little bit more current eventsy kinds of things, talking a little bit more about politics. Obviously, we've done some of that on the show. But I think Substack will make it easier for us to sort of do that piece and link it to the podcast without putting all of that stuff directly into the audio feed. So for those of you that want a just the science, ma'am, kind of show where we leave politics mostly out of it, unless it's incredibly relevant, you can have that experience, and then we can also be doing some of the other not quite advocacy, but at least talking through what it's like to be a scientist in 2026.
Yeah, letting people know, what's going on and what it looks like when every single NIH director is either ejected or ejects from leadership structures. So
Yep. And when the NIH decides that actually what we really care about is chronic Lyme, and vaccines are probably not a thing anymore.
right. But
These
Lyme,
things affect us.
delicious, delicious,
Yes,
makes you feel
exactly.
better. No, no, and I would emphasize to our listeners, there were a lot of I statements that Kevin just put out there, and all of that is completely founded and accurate. Those of you that are listening know that Kevin and I have been doing this a while now, and Kevin has taken the vast majority of the production of this show. Basically on his shoulders and carried it through the last 10 years. So first of all, thank you, Kevin, for doing all that work,
My pleasure.
particularly the website that we are about to leave behind. I know how much time you put into that. I'm sorry that we are leaving it behind, but I hope that both for you and for the listeners, this becomes a more seamless experience, and we can, focus on the stuff we want to talk about, some of the science, some of the advocacy stuff, or some of the policy stuff. So yeah, thanks for dragging us through that transition. And I'm excited to go into 2026 with sort of a fresh feel to some of this.
Yeah, absolutely. And I think there's going to be a couple of hiccups with Patreon. One of which is just that there's a minimum amount that you can charge people. So for those of you that are supporting us on Patreon at sort of lower tiers, like $1 a month, those tiers are not available on Substack. And so I'm looking into other ways to like make that doable. So you can maybe give us like $12 at the beginning of the year and then not again. Like I'm still trying to sort that out. The truth is, like, we don't pull in much income for this, so it's just like a little icing on the top to pay for hosting and things like that. It's not a huge deal.
Yeah.
We do absolutely appreciate it, and we would like to sort of increase the amount that we are giving back to folks if they contribute. But it's been a small enough audience to this point that it hasn't been it hasn't made sense for us to put in that extra effort. I think that after making this transition, it will free up a little bit of that time to do that. The other side of me taking on all of the technical stuff is that sometimes, as with today, I kind of don't read the paper. I mean, I read it, I glance through it, but I don't do the careful background investigation that I might otherwise do if I wasn't dealing with all of the web BS. So, with that, Matt, what are we talking about today?
Yeah, Glaaaands, Kevin. So you're an immunologist, and I think one of the things
On my
that
better days, yeah.
I, I think one of the things. So first of all, before we get into that, one of the wonderful things about going back through Substack, because as you know, we had to sort of manually take all of the episodes that were previously posted and pull them over into the Substack feed, and so, and that's fine, but. The number of show notes at the beginning that emphasized just how much we were drinking while doing the early episodes of this.
Yeah.
I sometimes I worry that we're missing that je ne sais quoi of like just accidentally rambling off the side of a topic.
Yes.
We may have to have some periodic episodes where we stumble ourselves back into that. Although I think that with children, they will be recorded at different times of the day than they used to be recorded at. So,
well, I just
that idea because I think that at times it added, at times it definitely subtracted. There's no doubt about it.
I mean, it added to my enjoyment personally. I did just purchase a mini fridge. It's not in the office yet, primarily for half and half for my coffee. But I would not be averse to storing some beers in there and cracking one open at 2 p.m.
That's an interesting
when we record. I think we should. I think we should definitely consider that.
Yeah, I think so too. So in grad school, we used to have a drawer of great and/or terrible results, and that drawer had liquor in it. And so, depending on whether your results were great or terrible, you were either celebrating or pretending like the last 3 days of your life weren't just wasted. So, I think that maybe we, I think maybe we could bring some of that energy back.
Definitely. And I just, on that note of liquor, this was my present for getting my faculty position from my previous mentor, Vanya. She got me these rocks glasses. I don't know if you can see. They've got the Darwin tree that he drew in his notebook, and then on the other side, a sort of like modern tree of life. There, and it's a pretty nice rocks glass. So
it's a nice rocks glass. Do you ever wonder if like
I would have scotch
Darwin
on the rocks and do audio immunity.
Yeah, I'm for that. Do you ever wonder if, like, so you're Darwin, right? And you're wandering around the Galapagos, and you're on and off boats, and you're talking with folks, and you're looking at stuff, and you like jot this thing down in your notebook, right? And then later, as you become more established, as people start taking you seriously, people are going back through your notebooks and stuff, and they pull this one image that then like propagates ever. Do you think he ever was like, I wish I spent just a little more time drawing that one thing, right?
I think he had plenty of other things on his mind. And he jotted a lot of shit down in his notebook. Like, have you seen the thing where he like is talking about like whether he should propose to the woman he was seeing, and it's like a pro and con list.
Yeah.
And it's like, better than a dog.
Right.
We'll have less time for like doing science. Like the man was prolific, like probably a genius. Although, you know, I think that term gets thrown around sometimes a little haphazardly, and like other people were coming up with similar ideas. But if you go back, as I did, this is probably like 15 years ago, and you read The Origin of Species, it's like we sort of understand the end result and the combination, what's sometimes called neo-Darwinism, of like the synthesis of Mendelian genetics and our understanding of molecular biology and evolution. Like those things together are really the foundation. But if you go back and you read The Origin of Species, and you just see like a huge chunk of that book is like, okay, how could things have gotten to islands? Let me throw some seeds in a bathtub and see how long they will float without like, and still be able to germinate. Like, and is that a plausible way? Like, it wasn't just the observations. He was doing experiments. He was like meticulously cataloging pigeon breeding, you know, to like see like if the changes that he was witnessing were possible by a lot. I mean, it's just like, it's a monumental work.
Yeah.
I think
it'd be kind of fun to do that kind of project with absolutely no platform to build off. And you're just like, I have decided that I'm just going to take this idea and I'm just going to spend my life doing stuff around that idea with really no background information as to how to approach it. Like it is a fascinating read, definitely worth going back and reading it beyond just the evolutionary component, as Kevin was just mentioning. That was actually one of my biggest, God, it was an undergrad project where I was going back through and I was annotating components of the different versions of The Origin of Species because it developed over time.
Yeah.
And so it was
it was like 30 years after his journey to the Galapagos that he wrote it, right? Or that he published it, I guess. He was writing all of that time.
There were iterations of that too. And so I was like cataloging, boy, this is nerdy. I was like cataloging what had changed through the various versions with a faculty member at the time that was going to the Galapagos every year. But anyway, yeah, it's a good read. It's worth it, even as a sort of general science and history kind of thing. So
yeah, Glaaaands. You were talking about Glaaaands.
Glaaaands. So there are some things that stick in people's heads, I think, or into the lexicon, and they just really never budge. And one of the things that I think I always found very frustrating as a budding immunologist is the inability of the medical profession sometimes to correctly classify what a lymph node is, right? Because I have been to so many primary care appointments where somebody feels for my glands under my neck. And that's not a thing. I mean, there are glands in and around your neck, and those things are
That's not what swells.
Glands.
Yeah.
They make your saliva. That's not what's swelling. Your glands don't swell when you get sick unless your salivary glands are infected. And there are things like that. and they're bad. They suck. But I picked this paper by and large to help us draw the distinction between what a lymph node is and what a gland is. A lymph node is a place where your immune system and your adaptive response specifically sets itself up, basically patrols the periphery, watches the periphery for signs of infection, and most importantly, perhaps, filters stuff that's going through your lymphatic system, right? So we've talked about this on the show before, but by far, I think the best way to think about a lymph node is your blood, as it's leaking out into your tissue, which is intentional, is feeding all of the cells that it leaks out to, right? It's leaking out with nutrients, right? All of the things, hormone, everything that your cells need to respond correctly to its outside environment and keep its metabolism going. These things are being sort of leaked out from the blood. As soon as that fluid volume is outside the blood, it becomes lymph, right? Or we call it interstitial fluid for a little bit. It becomes loose, right? But basically, what's happening is your
Loose
cells,
fluids.
yeah, loose fluids. Your cells are bathing in fluid. It's not that there are some vessels and they control all of the liquid, and then your cells are dry on top, right? Everything is kind of in a bit of bath of interstitial fluid. That fluid has to go somewhere, right? It gets recycled. That's why your lymphatic system is an open system, not a closed system, because the stuff that needs to get recycled sort of passively drains back into your lymphatic system. It goes from sort of outside. I'm like raising my arms out here. It
Yeah, I
goes
can't
from
see
your
it.
fingers back through your arms, right? And eventually, what happens is all that fluid gets recycled back into your left subclavian vein, which is directly connected functionally to your heart. So all that fluid that was leaking out of your vessels is now finding its way back into your blood circulation. Great. Problem with that, of course, is that if you get an infection in your finger, right, the bacteria that seed that infection are also in your interstitium, right? It got through the dermal layer, and so now it's hanging out underneath. And so that bacteria, there's no reason that it can't also find its way into that fluid that's bathing your cells. It can't find its way into the lymphatic capillaries that are going to eventually take all that and recycle it back into the system, right? Which means you need to filter it. It cannot just go back into your blood. The way that we filter
Famously, bacteria in your blood is a bad thing.
sepsis. Yes, we don't like sepsis. Sepsis is bad, it kills people. And so you need a filter somewhere between your finger and your heart, right? Your lymph nodes are that. They are the filter. You have macrophages that sit in your lymph nodes. They sort of monitor all of the things that are coming through from the upstream lymphatics. And so by the time the lymph flows through your lymph node, hopefully it is clean. So when that fluid gets into your blood, you're not septic, right? So that is the primary, I would argue, the primary component of what a lymph node is and what it is for, right? And also, if all of that fluid is draining from all of those distal places in your body, it means that what you're doing is you are centralizing dangerous things into this filter, right? And you can use that for your adaptive immune system to sort of find what is going on out in the body without having to patrol everywhere and then send support, right? So, what we think of as sort of the primary function of immunologists' sort of centralized view of what a lymph node is actually really able to happen as a byproduct of the fact that these lymph nodes are filters and they can centralize things, right? That's a lymph node. Secondary lymphoid organs all sort of operate that way, right? The spleen is doing functionally the same thing, but it's trying to capture stuff from the blood. It's serving as a filter, right? Because nothing is perfect. You do get stuff that finds its way into your blood. That stuff needs to be dealt with quickly. Your liver does some of this as well, but your spleen is a specific lymphoid organ that uses that filter function again to centralize antigens and respond in kind. That is what secondary lymphoid organs are. They are not glands. Glands secrete things. Lymph nodes do not secrete things. Lymph nodes swell because your cells are replicating rapidly and they're being recruited to that site, and those cells will eventually act locally, and they'll also go to the site of infection and keep you safe, right? Your glands are producing things like salivary glands, like mammary glands, right? Milk production, like the glands up near your eyelids, your lacrimal glands, right? Those are producing your tears, the aqueous phase that sort of coats your eye, right? So these two things are fundamentally different. Glands have a specialized structure. They have obviously the cells that produce the thing that you're going to produce, right? And they also have, because they're sort of like, what are they called? The bulbs. You know, when you have a baby and you have to like suck snot out of the baby, right? This is
Yeah.
actually a really good way of seeing how far we've come. Like this is the thing that I'm coming up with. So you have those like snot sucker outer bulbs, right? Bloop.
I mean, we always used the one that had the long tube
right,
that you stick the toilet paper in and you, yeah.
Yep, you're welcome, everyone.
If you haven't had a child,
Yeah.
if you don't know, sometimes because babies are useless,
Yeah.
their noses will get clogged and they don't know how to breathe from their mouth. So they can literally suffocate when they have, like I currently do, a massive amount of congestion, which they have frequently. So you literally have to like suck the snot out of their nose for them. Otherwise they won't breathe. And I don't know why you brought this up.
Because
I'm confused
about
the relationship of
the
that.
shape of those bulbs is actually what the shape of the end of a gland looks like, where you have this sort of contractile structure. And so the cells, and if you're on YouTube, you can see the cells that actually produce the thing are here. And then there are these
around the internal part,
around the outside. And then that's all connected to the tube work, the ducts that are actually going to bring the thing to the place where it needs to go, right? And so those contractile cells do this. And they squirt the stuff out, right? And that is what your salivary glands are doing, right? You are actually, when you start salivating, those salivary glands, those stromal cells, MECs we'll talk about, are actually compressing the cells that are producing, and so whatever fluid is there is being pushed out into your mouth, right? Same is true of mammary glands and lacrimal glands and those sorts of things, right? If you start to tear up and you start, making more tears because there's something there, there's an active squeezing process that sort of brings that out, and that'll become important as we look at the paper. So anyway, that is my high horse. Glands are not secondary lymphoid organs. Secondary lymphoid organs are not glands. You can have immune responses at a gland, but that's because the gland has something going on, right? Now, that does not mean, however, that the immune system doesn't live around glands. Of course it does, right? Because it's everywhere. And
if it's in the brain, it's in your glands.
Right, exactly. And this gets to our, I think, theme of season 2, which is, hey, your immune system is doing something cool there too. So this paper, which is adenophages are an atypical macrophage population in exocrine glands sustained by ILC2-derived GM-CSF, is a paper about macrophages being in those areas.
Query whether they're actually macrophages, but we'll get to that.
Okay, and doing something interesting, right? So they're doing some unique biology that allows this system to function. So before we get into that too much, and I'm going to close that F31 application. So we have our first author, Friederike Westermann. And our last authors, Elaine Emerson and Burkhard Becker. And so this is it's honestly, I would argue, a pretty good piece of work. I think that we are going to be describing basically a triad of cells here. And they're all actually in the title. Actually, no, one of them didn't make it. So certainly we have these adenophages. We're going to be talking about the macrophages that surround these glands. We're going to talk mostly about salivary glands, although we will sort of gut check ourselves in mammary glands and lacrimal glands. We're also going to talk about ILC2s. So these are innate lymphoid cells, 2. And we'll
version 2.
Version 2. And so these are going to be really important in the induction of these adenophages, like how do they get to be there? And then finally, what we'll do is we'll talk about the impact that all of these cells have on that sort of contractile function of the gland. So,
and I should maybe mention, because it's in the title also, this exocrine gland, as opposed to an endocrine gland, which is something like your pancreas, right? Which is producing, so the things that Mac was talking about are all fluids that glands are producing that are to some degree going to be go outside your body, right? Tears, milk, saliva, all of these are like getting outside the epithelium and into the sort of topologically exterior space of the body, as opposed to something like an endocrine gland, the pancreas, pituitary gland. These are producing hormones and things that are regulating the internal state of your body, as opposed to secretions that are going outside. So, it took way too long to get there, given my inability to speak. But
yeah, and I know at least one of our regular contributors will get this, but I would say that the one thing that Latin did in Magistra Sugarman's class was teach me the difference between endo and exo. I will say, yep, that was, yep, very, very little did I take from all those classes where I consistently got an 86 for years and years and years. Yep, but that's one of them. So, let's just jump in. The first thing I want to talk about a little bit is what an ILC is. And this is actually something that I don't know that we've talked a ton about in this podcast. And the reason for that is because when you and I were training, it was starting to be a thing, but it wasn't really a thing yet.
No.
So, we have different lineages of immune cells, right? So we come from, when we're talking about immune cells, and this is not entirely true, by the way, but mostly what we're talking about are cells that are derived from hematopoietic stem cells. Now, I will say that there's an entire biology of developing fetal immunity that we're leaving behind here, and that's actually really important because, and they sort of gloss over it a little bit in this paper, but it turns out that as a developing fetus, your immune system basically seeds your entire body with a set of cells that are phenotypically independent of the adult version of those cells, right? And they're going to be there forever. Many of them are self-renewing. They become a really important component of your overall biology and your immunity. But when we're talking about immunology, we're frequently talking about adult immunity, and when we're talking about that, we have different lineages that come out from that hematopoietic stem cell. One of the most discussed is the lymphoid lineage, right? And we discuss it a lot because adaptive immunity is sort of sexy, right? It's the specific part of your immune system, T cells and B cells and firing off antibodies and like all of those things, which is fine. Not so long ago, less than 2 decades ago, people started realizing that there was a component of lymphoid cells that did not have the receptors that we traditionally associate with B cells and T cells, right? And we've talked about a lot the recombination of receptors that happens in order to make a T cell receptor and a B cell receptor. All of these are things that make these cells capable of identifying a specific antigen versus something more generic. But there are cells that come out of that lymphoid line that do not turn on RAG, the recombinase activating genes, right?
Yeah, maybe I wonder if it'd be worth just briefly saying, so just because you went on the rant about lymph nodes,
yeah,
one might say like lymphoid means that it's in the lymph or something. That's not the distinction. When you go from a hematopoietic stem cell and you start differentiating, all of the sort of cells of the immune system and also red blood cells come from these hematopoietic stem cells. And there's a couple of branching points. So one is the branching point for erythroid cells. So that's your red blood cells and your platelets. Then there's the main two branches that we often think about in the immune system are the myeloid lineage. That is all of your other white blood cells, the neutrophils, the macrophages, the dendritic cells, like all of that stuff. And then there's the lymphoid lineage. Typically, or at least early in immunology, were the T cells and B cells. That lymphoid lineage that branches early on are generating the adaptive immune system. That's what Matt's talking about here. It turns out that there is another branch point after the myeloid lymphoid branch of differentiation. Within the lymphoid branch, there's this other branch that generates other innate cells that don't have T cell and B cell receptors.
Yes. Exactly. So yeah, when you're talking about the generation of B cells and T cells, those B cells and T cells come from a common lymphoid progenitor, or CLP. It turns out that those CLPs do certainly turn into T cells and B cells, and also these innate lymphoid cells, right? Now, interestingly, these cells have flavors like B cells and T cells do, and specifically T cells. I think that although I would argue that this is silly, people generally think about polarization specifically in terms of T cells, right? And I think it's just the way that the field has developed. I would argue
Right.
you probably find similar polarizations in B cell responses, but I think it just looks a little different, so we call it.
Well, and macrophages and DCs too. I mean, it's,
Right, exactly. But we have very, very clearly defined types of T cells, especially helper T cells, which are going to be, the things responsible for stimulating B cells, helping macrophage, all of those sorts of things, right? And we tend to classify them by the kinds of cytokines they produce, right? Those cytokines have a direct effect on what those T cells are going to be capable of doing, whether they're going to be more allergy focused versus extracellular pathogen focused versus wound healing focused even, right? Like all of those things are sort of dictated by the kinds of cytokines you release after your T cell receptor has been stimulated, right? So we call those polarization states. We have a bunch of them. In the T cell world, we have Th1 cells, which are, again, those sort of extracellular pathogens, the Th2s, which are tend to be a little more allergy focused, Th17s, right? We have a bunch of different sort of versions.
We skipped 3 through 16 for whatever reason.
Yeah, so yeah, and the reason for that, for those of you that are wondering why did we actually
Matt,
do that?
Don't say BJ's poem, please.
Yeah, I'm not going to. I was a virgin T cell. Anyway, Th1 cells were the ones they discovered first. Th2 cells were the ones they discovered second. Th17 cells produced IL-17 because that's what immunologists do.
Yep.
They
sure do.
That we've started. I've decided that we're just going to call it something different, but so be it. Here we are. We are describing the cell.
Didn't somebody try to make a Th3 and everybody was just like, no,
everyone's
we're
like,
done.
Nope, we're done.
We're done with this.
Actually, yeah, it was a good start, but after 2, we got exhausted.
Yep.
Okay, so we have these innate lymphoid cells, and it turns out that they polarize similarly to the way that T helper cells polarize. And so there are T cells, or there are innate lymphoid cells that sort of specialize in the production, for example, of interferon gamma. And interferon gamma is one of the major effector cytokines from Th1 responses, for example, right? And the same is true of other innate lymphoid cells. And so we sort of categorize them the same way in some circumstances as we categorize T helper cells. So an ILC1 is an innate lymphoid cell that has a tendency to produce the cytokine profile of a Th1 cell. An ILC2 is producing the cytokine profile of a Th2 cell, right? Now, importantly, it's not that those cells are getting T cell receptor signals, right? And that's the difference between these two things, right? ILCs actually get stimulated in other ways, right? And frequently they just serve as signal amplifiers. So they're there, they receive some inflammatory signal. They take that inflammatory signal and they signal amplify into the polarization state that they are designed to polarize towards. Now, interestingly, they can also respond to the nervous system in some cases. And so you actually see this later on in the paper, and I don't know if I'll get to it, but they can actually respond to signals coming from nerves and produce those same cytokines. So it is a way to take the polarization flavor of these TH cells and sort of functionalize that in a way that it does not have to be antigen specific. It has to be environmental specific. Does that make sense? I don't know.
Yeah, I think so. I mean, I think it's worth. So, all of these things are. How much I want to get into the emptiness of this. All of these categories are made up, right?
We're gonna go on Kevin's bin rant. We get it once every 3 episodes.
At least. At least. It's just it's a constant part of my thinking now, and it just keeps coming up. Because the more that we try to define, my major problem with this paper is just this idea that we're going to give a brand new name to a cell that is like a bunch of other cells, but not quite exactly like the cells that we've seen before. So we're going to call it an adeноphage, but it's phenotype is just all over the map. So that's my main problem with this paper is it's just we are so desperate for categories that we are going to give a brand new name to a cell type that's anyway, doesn't matter. But in terms of this, there are a lot of cells that can produce cytokines of various flavors. We have labeled some of them adaptive cytokines. We've labeled some of them innate cytokines or inflammatory cytokines. ILCs are a type of cell that produces these adaptive cytokines, but not in response to a rearranged VDJ generated receptor. They're part of the same signaling apparatus that is going to promote a certain polarization of an immune response depending on the kind of infection or tissue state that needs to be resolved. And they do that. They do it in a slightly different way, and they have different markers. They have different CDs on their surface. So we call them something different. But yeah.
Yeah. So
don't get too married. Don't get too married to your bins.
Yeah, get out of here, bins.
Buddhists of the world, unite! No, yes. Sorry.
So this paper is actually about GM-CSF.
Yes,
at the end of
absolutely.
the day, this is a paper about GM-CSF. So GM-CSF is a cytokine that for a long time has been used to help differentiate macrophages in a dish. So what you do...
Do you know what it stands for?
Colony stimulating
factor
is the end, but the granulocyte monocyte, yeah. And so you're pulling basically you pull blood, right? If you pull peripheral blood and then you pull sort of the innate, that monocyte component, what you can do is you can drop it in a dish and you can add this colony stimulating factor. And what happens,
colonies.
Right. And what happens,
granulocytes and monocytes, it turns out.
Yeah, those cells get stimulated. And they turn into a highly phagocytic macrophage-like cell that tries to eat the bottom of the plastic, and they become very hard to get off the plastic because they're
and if you do this in mice, you take bone marrow from mice and you dump some GM-CSF on them, and they become macrophages also. And it is a nightmare to get them up off the off the bottom.
Lots of trypsin. So this is a cytokine that it's one of those things where we actually, I don't know that we ever really learned all that much about it in vivo, but it's a very useful tool in vitro because you can create macrophages in a dish and then study the function of those macrophages. Now, there have been questions for a long time about the fidelity of those macrophages and their phenotype to what you would actually see in vivo. And I imagine that that's part of what this project is based on. Like, what is actually the in vivo role of GM-CSF, right? And so that really brings us to figure one. So if we go to figure one, figure 1A is a reflection of a reasonably straightforward genetic model tracking GM-CSF. So importantly, there are two components to this genetic model. Basically, what you've done is you've taken the GM-CSF promoter and you've tacked onto it two things. One is the expression of Cre recombinase, and the second is the expression of functionally GFP, right? Green fluorescent protein. So two things happen when you drive GM-CSF. The first is that you produce green fluorescent protein. The second is that you produce Cre recombinase. So what you can do is you can take that animal and you can push it into a model where if Cre recombinase is expressed, all cells turn red with red fluorescent protein. So I think that they're using the AI for the AI14 model. Is that right?
I don't recall.
Doesn't actually matter. Yeah, AI14. So basically what's happening in these mice is cells that drive GM-CSF produce Cre recombinase. That Cre recombinase turns those cells red because they are expressing now red fluorescent protein. And also they turn green because they are expressing green fluorescent protein directly. Here's the important part. The red stays on, right? Because the Cre recombinase has actually caused a somatic shift, right? And now it's just...
Yeah, you've rearranged DNA to turn this thing on permanently. So any cell... And it's... I think oftentimes we sort of elide this fact because it's just like so fundamental, but it's the cell and all of its descendants.
Yep.
So it is the lineage of cells that comes from any cell that has turned on GM-CSF will stay red. But as you said, the green is a transient reporter. So it's only on if GM-CSF promoter is actively being transcribed.
Right, if it's on right now. Right?
Yeah, so yellow cells are expressing GM-CSF at the moment.
Yes.
Red cells have at some point in the past made GM-CSF, but are no longer producing it.
Yeah, or a progenitor made GM-CSF.
Right, yeah, exactly. Right. Exactly.
So, yes. So they did this, right? So you build this system, you wait a while, and you ask the question, which cells are red? Which tells you which systems basically have expressed a lot of GM-CSF in the past. And my guess is that when they did this experiment, they were probably looking for lymphoid organs. My guess is that probably they would have been looking for things like lymph nodes and spleens and skin, maybe gut, right? Those kinds of things, barrier organs, that kind of thing. And in reality, I think the most interesting thing that pops out of this is the highest peaks come from the salivary gland and the lacrimal glands, right? A huge percentage of the CD45 positive cells that come out of those two glands have expressed GM-CSF.
And the skin and the sex cells for some reason. Like, what's that about?
Yeah, I don't know. Not sure. Yeah, I think an interesting follow-up would actually be reproductive organs and how
it
impacts
the testis and the uterus are popping out like crazy.
Yeah.
But lymph nodes and spleen are just nothing. Thymus, nothing.
Yeah, exactly.
Bone marrow, nothing.
Not really using...
Wild.
Not really using it. So yeah, I mean, most Nature Immunology papers come out from an interesting discovery, and I think that this certainly qualifies as that. And so then, the obvious question is like, okay, well, what happens there, right? Why are those organs producing so much GM-CSF? And so they go to a time course, right? So they've got this unique system where they can say, And I wonder if they put the GFP in afterwards to do this directly, but it'd be interesting to know from the authors which came first. But it's incredible foresight if they did both those things at once.
But
So,
here's my other question. This is another one of those like science process questions. Like, who when you're quartering a mouse, you're like looking at all the things. Like, who pulls salivary glands?
yeah, it's a good point.
Like,
So here's what you can do. So you can get fluorescence. Uh, I'll bet, I'll bet that they put these things in an IVUS system. I'll bet that they took the mouse,
and the
they
mouth
sprayed
lit
it
up,
out,
and they were like,
and
"Oh,
yeah, exactly, and the mouth lit up, and they're like, huh. Because they also look at sort of oral microbiome development over the course of this.
right,
I wonder if they had a mouth thing that was weird, and they're like, oh, I wonder. So
mouth thing that was weird."
yeah. So what they did was they took this system, which beautifully allows you to identify both the current and previous production of GM-CSF. They go into those salivary glands and they say, okay, well, when do you produce it? And they, I'm sure, looked at adult and old mice and they're like, wow, no one's producing GM-CSF anymore. They just did before, right? And so then they went back to, just postnatal, right? So at P0, there are very few cells that have ever produced GM-CSF, but by day 20, almost 20% have, right? And you can see that turn on. You can see the percentage of current GM-CSF producers occupy a pretty high percentage of the overall population. So interestingly, by P30, postnatal 30, so 30 days after the birth of these animals, most of the cells are not producing GM-CSF anymore. Some still are, but most of them have not. And by adults, none of them are, right? You're done. So then the question becomes like, who's producing all of this GM-CSF? And so they went to high dimensional flow. And here's the thing that I really appreciate. They didn't make a big deal out of this. This has sort of become standard at this point.
Yeah, right.
They've got a high dimensional flow panel. They assess the lymphocytes. They put it onto what one would assume looks like a UMAP from single cell RNA sequencing, and they just basically said, nope, we use the flow the way that we're using high dimensional data now, which is, I think, the right approach. And it turns out that there are a pretty high fraction of ILCs in the salivary glands. Like, this is not, if you did flow on a lymph node, for example, you would never, ever get this fraction of ILCs, right? Like, this is way, way high. And you've got two kinds. You've got ILC1s, which again are that sort of TH1 style ILC, and you've got ILC2s. And so if you ask the question, who is currently producing versus has produced GM-CSF, it turns out that a lot of the ILC1s have produced it in the past, but the ILC2s are actually the ones that are producing it currently, which honestly, to me, brings up an ontogeny question, which is, can ILC2s turn into ILC1s after they're done producing GM-CSF?
Yeah, or like, are they all like producing GM-CSF early in the lineage, and then the ones that are differentiating into ILC1s are the ones that turn it off?
Yeah.
And the ILC2s just keep going.
Well be.
Just quick clarification. The other big population of cells here are NK cells. NK cells are also lymphoid derived, right?
Yes.
Okay.
Yep. So then in D, I find D delightful. And here's why I find D delightful. I find D delightful because they're like, let's do a single cell RNA-seq experiment. Right? Because that's what you do. We find these things now, and the first step is that we throw $30,000 at a single cell RNA-seq experiment of all of the lymphoid cells, right? And they took those RNA-seq experimental data, and they said, okay, where are my cells? And they classified those cells, and then they said, we are going to use this gigantic data set to show gene expression of one gene.
One gene.
So what you're seeing there looks like it could be a PCR.
Yeah.
And in reality, it is actually an RNA-seq experiment showing that GM-CSF is really just produced by ILC2s, maybe a tiny bit by NK cells or other ILCs, but realistically, it's produced by ILC2s. So what they've got here is they've got a clear signature of GM-CSF coming from the salivary gland that seems to be targeted towards these ILC2s. And I think that that's pretty solid. Then they go into the macrophages, right? And they say, what macrophages do we see functionally, right?
And the reason for doing this is because historically GM-CSF used to drive macrophage differentiation. So like there's a logic here.
Yeah.
To move from the ILC2s to macrophages.
Yeah. No, if you've got a GM-CSF function, it's an obvious move. So makes tons of sense. If you look at macrophages, basically what they do is they use a classic macrophage characterization scheme, which is MHC2 by CX3-CR1. They describe two major macrophage populations that are CX3-CR1, so that's the fractalkine receptor positive, and then they're either MHC2 positive or negative. But what they point out is when they look at the salivary gland macrophages, they actually see a third population developing over time, and that is a fractalkine receptor negative MHC2 positive population that's coming out of their gates. Now, you mentioned not being exactly clear what that population is, what the ontogeny of that population is, what we should call it.
I mean, I believe I mentioned emptiness is what I mentioned.
Yes.
You inferred all of those things.
The void. So staring into the void, it's not clear.
The inherent boundlessness of all things.
Right.
Is what we're talking about here.
Yes, exactly. So, these follow many of the macrophage rules. They follow a non-canonical association with these two markers. They seem to be, they do find them in other glands later.
Question,
go ahead.
Do dendritic cells express fractalkine receptor?
My understanding is that they can, and they do talk about CDC3s, which I know very little about later in the paper.
This to me, like we talked like years ago, because this was a hobby horse I got on in grad school about plasmacytoid dendritic cells. Longtime listeners will likely remember rants of this nature, and all of my like current obsession with boundlessness originated there. Okay,
you were mad.
I was mad because PDCs, dendritic cells, are things with dendrites. Plasmacytoid essentially means spherical. These are cells that are spherical cells with no, it just makes no sense. And they're called that for dumb reasons we won't get into. I feel like this is a similar thing, where it's like, this is a cell that is not, like, it's kind of almost the reverse. It's a cell that, like, it looks like a dendritic cell that is really phagocytic. So is that a macrophage that's DC-like, or is it a DC that's macrophage-like, or is it just, like, should we just not try to characterize these things?
Have our bins broken?
Yeah.
So I will say that that the name itself is closer to what is probably the reality, which is a phagocytic cell that sits in glandular tissue. I actually think that the name itself is reasonable, whether we call it a macrophage, whether we call it, some hybrid of these things. I think, these are cells that express bona fide markers that macrophages generally express. They express it a little weirdly. Their phenotype is a little bit different. They do retain their phagocytic capability, which we'll talk about in a second. Regardless, they find them, right? They report them, and they find that if you do not have the GM-CSF protein, right? If you can't produce the protein, so GM-CSF knockout animal, and you look in those macrophage populations, you lose the adenophage bin, right? So by day 30, it looks like a fairly significant fraction in a wild type animal. I don't know if I can pull the actual numbers, something like 10%, 10% or so of total CD45 positive cells would be these adenophages. But if you knock out GM-CSF, basically that number is functionally zero. And so they show that as a percentage of CD45, and also they just like count the number of these things in the salivary gland itself, which is actually a lot, surprising amount. They're doing a good job isolating these cells. I guess that's the point. It's hard to do stuff like that. So then you can ask the question, well, we saw GM-CSF in the ILCs, ILC2s. We see the loss of these macrophages. Is the loss of the macrophages dependent on the loss of ILC2 produced GM-CSF, right? Because that has not been tied up. And so what they do is they go in and they say, okay, well, we're going to knock out GM-CSF just in the hematopoietic lineage, right? And yes, you lose those macrophages. So whatever is driving macrophage differentiation is a hematopoietic derived source of GM-CSF. And you go one step further. It turns out that there is an ILC2 driver. And interestingly, I had to go back and look what this protein does. It's actually a neural receptor functionally that ILC2s produce, which is kind of cool, right? We talked about how these cells can be responsive to the nervous system. But it is specific to ILC2s. And so what they do is they basically knock out GM-CSF specifically in ILC2s. And sure enough, that drops basically to essentially nothing. So what that tells you is that these ILC2s that you found by single-cell RNA-seq expressing GM-CSF, if you knock out that expression, you actually do eliminate this adenophage population. I imagine because they saw weird things in the mouth, the salivary gland, they probably wondered reasonably whether this was dependent on some kind of interaction between the mouth normal flora and, an immune response to that normal flora, right? Because you're macrophages, right? So it could well be, and it turns out that that's not the case. So if you get rid of normal flora through germ-free animals, right? So no bacterial colonization of the mouth. You still get GM-CSF production. You still get adenophage differentiation. So this seems to be an inherent function of the salivary glands, not...
this is developmental, not stimulated.
Exactly.
Yeah.
Yeah, that was more concise than the way that I was...
It's a cool... It's cool that you put negative data like that in a main figure. I feel like that was probably an expensive experiment to do because germ-free mice ain't cheap.
Yeah. So
zero phenotype. Quite wild.
So you have these cells, right? And you're thinking to yourself, what are these cells? What are they doing there, right? They're not responsive to bacteria. The germ-free animal experiment failed, presuming that, this is all kinetically linked, which it almost certainly is not. So what are they actually, and what are they doing? And so they go back and they do high dimensional flow, and they basically say, what do these cells look like, right? And so what you're seeing basically in figure 2 is
that they're dendritic cells.
Essentially, that the biggest clustering of cells that these things seem to be associated with are actually CDC2s, so dendritic cells. But nonetheless, we have a whole bunch of markers. That's according to these markers, Kevin. According to these markers.
Well, exactly, and that's exactly the point, right? Yeah. Is that we build these categories based on markers, and then what we actually do is we look at phenotype, because phenotype is what really matters. So,
which they do, which is good.
Yeah, no, totally.
So we've got these adenophages, and they're expressing a whole bunch of things. I don't think that we need to get into the binning of this. If you're a macrophage or dendritic cell person, go to the
figure 2 is worth looking at.
You can figure out how to do a flow experiment and find these adenophages. They've got lots of markers. Some of them are useful, some of them are not. It seems like, they express slightly more CD226 than most other dendritic cells that fell out of the plot. Fine. So then the question is, where are they coming from? Where are they coming from? Right? They actually find that a fair fraction of these things are coming from fetally derived lineages, right? So it turns out that there is probably a wave of fetal precursor seeding and production of it, which makes sense, right? If this is developmental, there's no reason to wait for an adult immune system in order to do that work.
Right, and so that's a non-hematopoietic lineage.
Yes.
Is where the cells themselves are coming from. We said the ILCs are hematopoietic derived. We know that because if we knock out GM-CSF just in hematopoietic cells, we don't get these cells. But then the cells themselves are coming from non-hematopoietic lineages.
Yep. And so, yeah, I mean, basically, I don't know that we need to go this hard into it. They basically said, these cells are, there's some fetal origin, and when it's not fetal origin, they're not developing the way that you'd expect these macrophages to generally develop. They're not going through the sort of traditional bifurcation pipelines that you'd expect here. The path that they seem to be choosing is a weird path, going through these MDP monocytes, which CDC3s go through, and they're kind of like off to the side and weird, So there's a niche developmental pathway here that these adenocytes or denophages are going through. And they talk about that in a couple different ways. They talk about some of the unique gene expression profiles that these cells have in comparison to all of these other innate components. Again, unless you're looking to do some flow and really investigate these things, I don't know how useful it is to dig into this in detail. But they do provide a sufficient roadmap for you to be able to find these cells if you were to carefully pull apart a salivary gland, right? So the bigger question, I think, and the more important question that Kevin has been alluding to is, what do these cells do, right? So we know how to find them. We know that they're there. We know they're dependent on ILC2. What do they do? And so what you've got in A of figure 3 is functionally just a differential gene expression plot, right? And so on the left of that plot, you have CDC2s, which is what they clustered with during that high dimensional flow. And on the right side of the plot, you have genes that are differentially regulated by adenophages or upregulated in adenophages. Again, I actually don't think that this is all that important. They show you a lot of those the genes that came up. You can see the heat map in comparison to other innate cells. And here's what I'll say. I've said a couple times now, it's not important. It is important to publish this in the paper. If you're identifying a new cell type and you're calling it something, everybody's going to ask you what the flow profile looks like. Everybody's going to ask you what the gene expression profile looks like, right? It is important to, put enough of that information out there so that people have an anchor into what you're talking
it's not
about.
important in the sense that for this podcast, like the details are not relevant.
Right, exactly.
Really.
Exactly.
The broader characterization, I mean, I think B and D of this figure, of figure 3, are really the strongest arguments against the things that I'm saying. So B is the gene expression profile of the, I can't remember if they filtered on the most differentially expressed genes or exactly how this clustering happened, but at least from what they display in B, these cells don't look like macrophages and they don't look like dendritic cells, right? Morphologically, they look like dendritic cells, not macrophages.
In C.
In C.
Yep, they kind of have that like roughly bally kind of thing. That's what a dendritic cell looks like under steady state, by the way. We talk about the dendrites with dendritic cells and dendrite is branch, right? It means branch. And so they have these protrusions that you can see morphologically, but under steady state, they've got sort of this high surface area ruffled appearance before they get activated. And they definitely have that.
And then in D, they're more phagocytic than macrophages, right? They're like wildly phagocytic. At least for this assay is like they've got these little particles that look like Staph aureus, and they're grabbing those. And then in E, they're doing antigen presenting like a dendritic cell. Like exactly. This cell is just all over the map in terms of its phenotype, right?
Yep. Yeah, that's right. And so then they ask the question, so what if you infect it? Which is fine, because I mean, you would want to know, right? Like maybe you struck out on and I imagine this experiment actually came before the germ-free experiment, but maybe you struck out on the germ-free animals, but you're still thinking to yourself, you've got this wildly phagocytic cell that seems to be able to present antigen. What if you hit with a virus that is sort of specific to the salivary glands, right? Like you get in there and like, do you mess with the phenotype? Yeah, so what they do is they go and infect with an opportunistic pathogen, Candida albicans, and they ask the question, does anything happen, right? Are you hampering immunity to this thing that, if it's essential for infections in and around the salivary glands, might show up? And the answer is nope, not at all. So what they do is they look for expression of a protein that Candida expresses early on in infection. They ask the question, does expression go down or up, right? Which would tell you that maybe Candida is flourishing a little more, right, or being suppressed a little more, and functionally absolutely nothing happens, right? So it seems to be that the presence or absence of these adenophages is completely irrelevant to the immune monitoring function that's going on in the oral cavity, which is cool.
For this one pathogen,
that's true. Yep.
So Candida, talking again about gross stuff with babies. Candida in the mouth is something, it's called thrush, and you sometimes see this with babies that are on antibiotics, yep. Which is basically like these white nets form in their mouths, and it's quite gross. But I don't know that this is the most physiologically relevant infection. I mean, the thing that's interesting to me about this phenotype, and given the location that we're seeing it in these exocrine glands, these are places like glands. Matt was describing the shape of them. They're these pockets where you can imagine it would be really easy for bacteria to like set up shop in these places. And so you can imagine that it's a very important site of immune control. You would want an innate cell in there that is quite good at cleanup, macrophage, and also quite good at stimulating adaptive immunity, dendritic cell. You might not want to wait for these things to drain to a local lymph node before being able to activate. Or maybe, they're activating the cells that are coming from the lymph node and have already been activated, and they're like helping to get this response going or keep it going.
Yeah. What would be a good pathogen?
Staph aureus.
Oh, interesting. Mump is a virus that infects the salivary gland and makes you absolutely miserable. So we might get to test it. So we'll bring mumps back because vaccines are bad. Make salivary glands great again. So
here's the thing, though, Matt, is that disease is caused by the environment, not by germs, and so
that's fine. So where are these things? If they're not important for containing thrush, are these cells sort of in physical locations with each other the way that we would expect? So right now we have a bridge between ILC2s and we have adenophages. If we actually look at these things, and to be honest with you, this is a little disjointed from the rest of the figure, but this is what happens at the end of publication. It turns out that the macrophages that you expect to be in that environment are close to the ILC2s that you expect to be producing the GM-CSF, and they're not necessarily physically in proximity with ILCs that are not producing GM-CSF, right? And so you've sort of physiologically tied these two things together in space, which for those of you that listen know that that's actually very important to me. Spatial context matters. So what are these cells doing then, right? And so, they finally, in the end of this paper, not finally, they do a good job all the way through this, but at the end of the paper, they say, okay, well, if this seems to be independent of immune monitoring, maybe there is a physiologic function that these cells are having on the function of the gland itself, right? And that gets all the way back to what is a gland, right? How does it actually function? And so they ask, I think, a completely reasonable question. If you look at these adenophages, what are they near, right? And so they find a couple things. The first is that they are near these MECs, right? And an MEC is finding the actual thing because I'm going to screw it up if I don't, a myoepithelial cell. Right? So a myoepithelial cell is that contractile cell that I was talking to you about before. These are the ones that sort of do the squeezing of the bulb. The next thing that they see is proximity to the acini, and those are the actual production cells, right? So if you're thinking about it, the macrophages are closest to the contractile cells and pretty close, but a little farther away to the cells on the inside that are actually producing the thing, right? So what they're starting to get a sense of physiologically is that you have these bulbs that squeeze out liquids into the duct, and these macrophages seem to be sitting on the squeezy part.
We're so precise with our language here. I love it on Audio Immunity.
It's best.
Close to the squeezy part. As an aside, this episode, I'm just like, I'm spoiled for episode titles here. Close to the squeezy part. I think the other one that I grabbed very early on, now I can't scroll back on my paper where I wrote it down. It doesn't matter. Bathing in loose fluid is going to be the other potential.
Those are good. So we've got these adenophages that are sitting next to, and FBs here, by the way, are fibroblasts. So that's kind of your just general stroma. That's kind of, honestly, a bullshit assessment, but that's fine. It's ducts. So the actual duct work that you're squeezing into, they're not near the ducts, actually. And that, I think, is very relevant. They're not next to the part that's like shuttling the fluid. They're next to the part that is producing and squeezing the fluid, right? So then the question becomes like, okay, they're next to these cells that are for contractile function, right? If we look at those cells in the absence of those adenophages, do they have changes in gene expression? So what they're looking at here, and this is actually interesting, what they're looking at are signatures of contractility, basically. So, how do these mechs generally function? What are the genes associated with that function, and how do those genes change if we don't have adenophages in the mix? And so what they do is they look at, again, GM-CSF knockout animals which do not have these adenophages, but they're looking now at the mech gene signatures, and what they're finding is that those cassettes associated, those signatures associated with contractility are all going down. And this is just a traditional RNA-seq experiment. And this is where it gets fascinating. I really, really like D. And I want to, whoever was the author that figured out D was very careful and rigorous. I really appreciate this because what you can do is you can stimulate salivation in mice, right? So you can collect apparently MIGs of fluid, by the way, like
mice are small. This is a large volume relative to their body.
It's a lot of fluid that they're collecting. So if you stimulate the salivary glands of animals, mice, you can collect it, and you can literally just weigh it. Now, what's interesting in D, 4D, is that by 20 minutes, There is no difference. They talk about this. 20 minutes after you start that experiment, there is no difference in the amount of saliva that you have produced. But what's fascinating is in the first 10 minutes, if you don't have adenophages and you knock out the contractility function of those mechs, you don't produce as much saliva. Now, interestingly, over the following 10 minutes,
You catch
production
up.
increases, right? And you can think about that in the physiologic function of how these glands actually work, right? Which is fascinating. The acini, the cells on the inside, are not affected by the loss of adenophages. There's no reason for them to be. They're going to continue producing saliva at exactly the rate that they would have been, except that these mechs can't squeeze. So what's going to happen is that fluid is just going to build up. They're
And
going to
drip
produce
out.
the same amount of
Yeah.
fluid. It's just going to slowly drip over 20 minutes. You can't actually pulse it in the same way, right?
Yeah.
So had they not done that kinetic time point, that breakdown between, the first 10 and the next 10, you might not have actually seen it. But I think that this is fascinating because it really pulls apart the exact physiology of what is going on here, which is that adenophages seem to be able to drive that one specific function of the gland. So anyway, I really liked that part. I really liked
Yeah. The thing that I think this brings up for me, again, thinking back to bins. I'm sorry to keep harping on this, but like, this is an immunology podcast. We've been talking about all of the immune functions of these cells. It turns out that like saliva is an important immune function, right? Like, as with many things in the immune system, the barriers between what is immune function and what is other bodily functions is very, very blurry. So like, if the point of these cells was to stimulate secretion, why would they need to be able to present antigen? Why would they be phagocytic? So, like, the idea when we talk about the immune system, they're managing homeostasis. Sometimes homeostasis is eating bacteria. Sometimes homeostasis is getting saliva out of a gland to either like wash away bacteria or just to like digest food, because that's a major function of saliva. So these cells seem to be jacks of all trades. When we label them adenophages, you are immediately associating in your mind their phagocytic function, their their garbage cleaning function. But it turns out that these are just cells in salivary glands that seem to have a diverse array of functions. And my point in harping on the binning thing is mostly just that if you allow the label to restrict your thinking in a lazy way, you are going to miss stuff. I actually really liked this paper. I've been sort of like playing devil's advocate here, but I think this is a really cool paper. They're really sort of like expansively characterizing these interesting cells that are doing just a bunch of stuff. And it's actually kind of funny that it's in Nature Immunology, because it like kind of undermines some of the other functions that they see. But
yeah, that's true. That's true. So once you're there, like now we have a function, right? We've started with the identification of a cell. We understand why they develop, how they develop, what they're dependent on. We understand what they impact. We understand what the physiological function is, right? We've sort of tied it up, and now the question is, okay, well, how relevant is this? We've been in the salivary glands the whole time, but we know all the way back from Figure 1A that the lacrimal glands, I think, is what they pulled, were also very high in GM-CSF, and we know that so the question becomes, do we find these cells, do we find similar physiology in these other exocrine glands? And the answer is yes. You can find adenophages in the lacrimal glands, in mammary glands. They are similarly dependent on GM-CSF. If you don't have GM-CSF from ILC2s, they also don't produce correctly. And then they look to make sure that those phenotypes are consistent in F and G. Basically, do these adenophages look like adenophages that we identified if we're going into these different exocrine glands? And they do. They're expressing functionally the same proteins, regardless of what the specific environment is that they're in. And then,
do they see them in mammary glands? Because I was curious about this because in Figure 1A, mammary glands have almost no GM-CSF.
yeah,
Past production of GM-CSF. The other major gland that they see is the adrenal gland, which is an endocrine gland.
Yeah,
not an exocrine gland, right?
I think they seem to, based on F, they seem to maintain the phenotype. Now I don't know what the fraction is. I guess actually in H they do show you that the fraction of it's no, they're there. They're there. But it's an interesting point. It's an interesting point. Yeah.
I mean, it would not necessarily be because, like, we've been talking about the developmental aspect of this, so mammary glands aren't going to develop until later. So maybe
it could
the GM-CSF thing that they were seeing, they were looking at an early time point, like before the mammary glands would develop.
That could well be. That could well be. But they do find them. They find the phenotype to be relatively stable. If you knock out if you knock out ILC2 produced GM-CSF, again, you knock out these adenophages in the lacrimal glands and the mammary glands. And then, importantly, if you go into a human being and you pull a salivary gland biopsy and you ask the question, the CD226, which they're going to use as a marker for these adenophages, which they identified back in Figure 2 when we glossed over it, you do find CD226 positive macrophages in the right spots in the salivary gland to suggest that this is probably the case in humans as well. And so J is really that end of a Nature Immunology paper now where you go, yeah, we did the whole thing in mice, and then we looked for this in one way in a human, and it seems like there's a pretty good chance that this is translational as well. Which is important, by the way. I'm sort of being glib about that. It is important to know if this is likely to function in a human being, but it is almost like the token human experiment at the end of.
And honestly, like I don't know that this tells us that. Like looking for one marker.
No.
Like it doesn't. But like, how much do you want to bet that there are like 5 authors on this paper that contributed that one panel?
Well, there's no doubt. There's a pathologist on here that has no idea what has been written in this paper. Yeah, I mean, there always is, right? Like, yeah. So that's fine. That's fine. It is collaborative science. That's how we do it. Not everybody has to know everything. So, yeah. I don't know. I liked it. I honestly did. I completely hear what you're saying about bins. There's a nice little diagram in Extended Data Figure 5. There's a cartoon in H that I think wraps up nicely what they see. I think it's pretty accurately represented. Immune cells do cool things in unexpected places, and it's not even always obviously immune related. Sometimes they're doing cool developmental things too. Like, yeah,
I would be shocked. I have to say, like, I also really liked this paper. I thought it was really great. My griping about bins aside, like, it's detailed work. It's, like, very, very thorough. I liked that they presented some negative data. I liked that they looked at non-immune functions. I would be shocked if there aren't immune functions here. I mean, like I said, like at the site of secretions, those are very, very attractive targets for infection to come in. And
like mumps.
Like mumps, yeah. I mean, lots of viruses, lots of bacteria. I was kind of surprised that they didn't look at staph, because that's a bug that likes to, maybe there's not a good model of salivary glands infected by staph.
So, yeah, I agree. I think this is completely reasonable. It's laid out logically. I think it's defended well. I certainly learned things that I did not know before this paper. There's a clearer understanding of what anatomy is and how it works, which I always appreciate when we can tie it back to that level. So,
yeah, totally.
Nicely done, all authors.
Yeah, and this is, we didn't say at the top, this is based out of the University of Zurich, which I feel like their email has immunology in the subject line. I didn't know about. My mom's from Switzerland, so that jumped out at me. I don't think I knew that Zurich had strong immunology program, but it's cool to see. I mean, yeah, cool paper.
Maybe a place to look as month.
Indeed. Send emails after the fact. Did you like our episode?
Exactly.
Can you give me a job?
This has been Audio Community.
Yeah. Remember I said we were going to leave politics out of it? So
no,
this has been Audio Community.
I will say that is the minimal amount that you get now, right?
True.
Yep.
True.
General snark is not something that we are going to scrub from these episodes, so if that rubs you the wrong way, sorry.
Indeed. I'm going to let you do the outro because my nose is just getting increasingly clogged up, and I feel like I'm going to stumble through it. So, Matt, take the outro away.
Well, but I don't know any of the things now. This has been Audio Community. We're no longer on any of the places that we were before. Now we're on Substack. Congratulations.
So we are going to point, audioimmunity.org is the baseline. We're going to have links that website isn't going away entirely. Eventually, we're going to be able to move the Substack to that domain. It hasn't happened yet, but it will. I think I will leave the Patreon up just like as an archive, and I'll make a post that says we've moved. So if you want to go there, you can. But if you want to subscribe and continue to support, head over to the Substack. I'll also, like I said, I'm going to look for possible platforms where folks that don't want to do $5 a month, which is the minimum that Substack allows, can still contribute. And the music at the beginning and the end is composed by Rachel Reinick. Hopefully healthy. We'll see you next time.
Sometimes you get lucky.